Hello everyone. I came here to tell a story about the order structure of proteins combating villains, Volume 1. So, once upon a time, in a very faraway place, there were molecules called proteins, very essential to all living organisms in the kingdom. So the proteins are consisted of a long chain of amino acids that are connected by a peptide bond. So the order in which the different amino acids are inserted determines the structure and function. So the chain starts folding until it reaches a final 3D structure that will exhibit its function. We know that the structure and function of proteins are encoded in the DNA. So one gene, we have one amino acid sequence, one protein structure and function. But it's not simple like that, because mutations can occur in the gene. So we have changes in the amino acid sequence, and so we have different patterns for a single protein. So for one gene, we have many protein variants, different protein structures, and different functions. So there are proteins with different structures and functions living in that kingdom, and everything was so harmonious in the living organisms in this kingdom until a threat showed up, the SARS-CoV-2. Yeah. So this virus caused the COVID-19 pandemic. And on the surface of this virus, there are the spike protein. So the spike protein has a region binding domain that will attach to the protein AC2 that is on the surface of our cells, so causing the infection. So mutations in the spike protein could change how well SARS-CoV-2 sticks to and does infect our cells. And some researchers that live in the kingdom systematically started to study the structure of different mutations, and while others studied what kind of antibodies that can combat them. So mutations in the gene leads to different proteins, different structure, and as I said, different function. So the approach that these researchers in the kingdom used, like I used, because I'm not just like a storyteller, I'm also a researcher. So is able to characterize the structure of the proteins according to their shape, by their mobility, and also by the mass. That's why it's our mobility mass spectrometer. So, offering the potential to correlate the structure of proteins, either in a compact or extended, to some kind of variation in the amino acid sequence. So, it's important to study the structure of the SARS-CoV-2 as well as other viruses, allowing understanding of the binding of the virus to our cells and also contributing to the immune response. So once we figured out how they bind to our cells according to the structure, we can develop antivirus, for example. So the proteins variant of the infection, thus the living organs of the kingdom were healed and lived happily ever after. Was this the end? Until another pandemic.